Okay, morning everybody. Thanks for coming early on a Sunday morning. So I'm going to start off talking about what we've done in terms of building up surveillance networks. This has been a large, truly global effort. So I'm presenting this, but many people have contributed to the work that I'll talk about. So, pathogens without borders. I mean, that's what we called this this session. So we're dealing with plant pathogens, crop pathogens, and they're real global travelers. They don't need passports. They don't need visas. They're extremely mobile. They're basically, I mean, it's the equivalent of the Zikas, the Ebolas, but on the crops that provide the food that we eat. So the way that these pathogens can move around the globe, there's a couple of really important ones. So wind dispersal is a key one, and certainly for the wheat rusts, it's possible that these pathogens can move certainly across continents, but they can also move even between continents. So the red arrows on this figure, it's a figure I got from a paper that James Brown and Mogens Hofmuller put out back in 2002, but it's a nice illustration. So the red arrows are wind-borne movements. They're all rusts. One's a wheat rust, one's a coffee rust, and one's a sugarcane rust. So you see that potential movement from Africa all the way across into the Americas, and in the case of wheat stem rust, southern Africa all the way to Australia. Now these very long distance events are rare. For the Africa to Australia, we believe it happened maybe 3 times in the last 60 years. So infrequent events, but they happen. The other mechanism is human-borne. So accidental travel or trade movements. And the blue arrows on the figure, they illustrate this. The one I would highlight is the blue arrow that goes from Europe, from France, all the way down to Australia. That was the introduction of wheat stripe rust or yellow rust. And the evidence suggests it was a farmer who went on holiday to France, and he took back with him an unwanted souvenir. And basically, wheat stripe rust is now one of the major diseases, economic diseases in Australia. It's causing, you know, hundreds of millions of dollars in losses to farmers. But these sorts of human mitigated movements, they're increasing at a frightening rate across crops, across different diseases. The world that we live in, it's ever more connected. The amount of travel, the amount of trade, and the pathogens are going along those routes as well. So we live in this increasingly connected world, and we're seeing an increasing number of food security threats. And it's really, how do we deal with this? I mean, we've got to have mechanisms in place. So I'm going to talk about a couple of examples of emerging transboundary threats. Most of the talk will be about wheat stem rust, but I'll also talk a little bit about maize lethal necrosis. So this is a new disease that's occurring now in East Africa. Both of these are killers of staple food crops. I mean, for maize lethal necrosis, it's in the title, yeah, the name of the disease, but wheat stem rust is also a killer. It's the most destructive disease of wheat. For both of these, large-scale surveillance and monitoring is critical. And by large-scale surveillance, it's basically, it's got to be global, or multiple continental scales. With wheat stem rust, we've really had a sustained effort since 2008. With maize MLN, we're just beginning to get started at the moment. So stem rust. Why is the monitoring so important? Why are we so worried about wheat stem rust? Okay, historically, it's the most feared disease of wheat. If conditions are favourable, it's capable of causing 100% crop loss within weeks. That wheat field you can see there is completely dead. About 2 or 3 weeks before that photo was taken, it was a healthy crop. The farmer was expecting 4 to 5 tons per hectare yield from that crop. He ended up getting 0. The other point about stem rust is it was considered the defeated disease. I mean, it was a major success story of agriculture. So, largely due to resistance breeding, and Dr. Borlaug was a key player in this, the resistance that was introduced essentially saw stem rust go from the most feared disease of wheat in the first half of the 20th century to 30, 40 years where it basically wasn't a disease of economic importance anywhere in the world. So it was a huge success story. That all changed with the detection of UG99 in East Africa in the late 1990s. This was a really unique race of stem rust. It possessed a package of virulence that we just hadn't seen before. And as a result of this, we were suddenly looking at a huge proportion of the world's wheat was suddenly susceptible to this killer disease. So this really put stem rust back on the map. Dr. Borlaug, who started his career in Mexico to address the stem rust problem, and then he put in the dwarfing genes and the Green Revolution occurred as a result, but it was stem rust was what he started to work on. And he ended his life, his career with this initiative against fighting stem rust once again. But he was really urging us, galvanizing the community, that we needed monitoring systems, we needed to be addressing this new threat. So maize lethal necrosis, just to give you a bit of introduction. This is a viral disease, but it's actually a combination of 2 viruses working together. The main one is this chlorotic mottle virus, but in combination with any of the potty viruses. Insect transmission, but it's also seed-borne, and we're beginning to learn that the seed-borne part is more important than we'd previously believed. First report in Kenya in 2011, and we've seen subsequent spread into neighbouring countries. So there are currently 6 confirmed countries in East Africa that we now know that maize lethal necrosis is present. So this one, it's a new one, and it's spreading, and it's a big cause for concern. I mean, maize is the staple crop for Sub-Saharan Africa. That lady there, that bucket, that's her entire harvest from her whole farm. So that's what maize lethal necrosis can do. It's a graphic example of just how devastating this disease can be. So stem rust. So back in 2005, we really didn't know very much what was happening. There was maybe around 10 sites in Africa where we knew this unique new virulence was present. But that was basically the sum of our knowledge. So what we needed to do at that point was we needed to build up the surveillance system. We needed to track the pathogens. The other thing with rusts is the classic quote from Dr. Borlaug is, "Rust never sleeps." It's continually changing, it's mutating, it's evolving into new strains. We also needed to obviously manage and disseminate information, improve impact assessments, and also try and put some early warning systems in place. So really I'll try and talk through what sort of progress we've made against these aims. So for the surveys, the surveillance network, so the yellow on this map is the distribution of wheat. It's the world's most widely grown crop, and it gives us 20% of our total calories. It's yeah, a really important crop. So when we first started, we were covering a tiny fraction. We didn't have eyes on the ground. We didn't know what was happening. Over the years, this is what we've been able to do. We've been able to put in place an extensive surveillance network covering 35, 40 countries, a large proportion of the developing world, wheat areas, and I think we've got to the point where it's probably the most comprehensive crop monitoring system that's currently operational. We've also been able to successfully track the pathogen changes. So we started off in 98, 99 with this single race in Uganda. The situation we're looking at today is we now have 13 races within the same genetic lineage. So we've got a whole family of these UG99 races. They've spread across 13 countries, so north to south in Africa, into Yemen and Iran in the Middle East. So the pathogen, it's mutating, it's changing, it's migrating. And these changes are still happening. We can't be complacent with these pathogens. So 2014, we saw 5 new UG99 races appear in Kenya. One of those new variants we also picked in Rwanda, Uganda, Eritrea, and Egypt from the same year. Now, okay, maybe it was there before, but this is still implying very rapid spread of these new races. And the problem's getting worse. I mean, these new ones are more virulent than the original one that came up, and there's serious implications with these new races for other major wheat growing areas. The other part of this, another part of the success is linking the surveillance to the breeding programs. So my colleague Ravi Singh, I mean, he heads up the breeding programs at CIMMYT. And basically, you know, we've been able to detect the important pathogen changes, take the most relevant races and use them in the screening nurseries, which Maris Ellis will talk about after me. And then the huge successes of the breeding program in terms of taking a strategy to develop a resistance that's based on multiple minor genes. So the issue with rust is if you have a variety with a single major gene, it's very easy for the pathogen to mutate and overcome that resistance. You put out resistance like that in an environment like East Africa, and maybe it will last two or 3 years at the most. But with this multiple minor gene approach, the expectation is that you would go towards durable resistance that would last for many years, and it would be race non-specific. So that's the sort of resistance that we want in varieties that farmers are growing. And the success is there. I mean, you can see here some of the numbers. These are the lines that go out in CIMMYT international nurseries that get distributed all around the world to our partners, and they're often the foundation for the country breeding programs to release new varieties. But you see a very high proportion of this, the resistance that we want in the material is going out to partners. So that's a huge success. And you can see here quite graphically the difference between a resistant variety compared to a susceptible. I mean, that susceptible is completely destroyed, it's completely dead. Kingbird has 4 to 5 minor genes, and it's a really key component of reducing stem rust populations in East Africa at the moment. And there are other similar varieties that are coming on board and going out to farmers. So we've had successes. We've done some very positive things, and I want to think about, well, what were the enabling factors? What made this happen? Critical focus was one, bringing everybody together. So UG99 was definitely that focus. Having enabling tools and technologies. Partners is a key one, and there's lots and lots of partners in this, and it's the whole spectrum. Continual engagement and community building. And here, I mean, Ronnie, the team at Cornell, I think has to take the credit. I mean, building this entire community. Prior to UG99, there were maybe, I don't know, probably maybe less than 10 scientists actively working on stem rust in the world. It was the forgotten disease, it's the success story. We've now transitioned to, annual conferences of several hundred people. I mean, this whole community coming together around rust issues. So that's amazing. That's fantastic. And the engagement with people. I've spent more hours than I care to remember walking through wheat fields with partners, doing surveys. But it's this continual engagement with our partners on the ground. The investments in capacity, both human and infrastructure. So investments in things like greenhouses, irrigation infrastructure, but training of young scientists, developing the capacity. I mean, now we're seeing increasing number of young dynamic scientists, increasing number of women scientists engaged in this community. And Maricelis, Jessica, I mean, they're great examples, very talented young scientists who are working to address these problems. Champions, I mean, Dr. Borlaug was a real driving force behind this initiative, but there are many others that we get to work with in the different countries. Some amazing people out there. So, tools, technologies. I mean, it doesn't have to be rocket science. Yeah, a piece of paper is a powerful technology. So what we did was put together standardized surveys. So having a standard survey that everybody was working with across all the countries was critical. We can translate that into electronics, so we can use tablets or smartphones to do the surveys. But basically we've got these survey teams across all the countries collecting data in a standardized way. Data management. So together with colleagues in Denmark at the Global Rust Reference Center, we put something together called the Wheat Rust Toolbox, basically a database, a data management system whereby we can easily get data into the system, we can do quality control, but then it's linked to a whole series of dynamic display tools, interactive maps, charts, graphs, so we can easily visualize that data. Information system, so we put together something called Rust Tracker. So this is a web portal where we can put all the information out to people. It's linked to the Toolbox, so those dynamic tools are all embedded within this. So as soon as data's in the system being quality checked, it automatically appears for people to see. And also we try and integrate pathogen information with host information in this platform. The pathotyping. So I've talked about, the races are changing over time. So the way we can detect that, it's through assays in the greenhouse that are done on seedlings. So this is pretty specialist skills, and we've been able to support and build up this global network to be able to do this. I mean, great support from the advanced labs. So here in the US, the USDA Cereals Disease Lab in Minnesota, the Global Rust Reference Center in Denmark, the AAFC lab in Canada. Great supporters, but also building up the capacity in countries, national labs like Ethiopia. New technologies, molecular diagnostics. So we've now got SNP assays for rapid diagnostics for UG99, high throughput SNP chips for population genetics. So this is really assisting us. Case of Ethiopia, 2013, we had this race, Digelu race, come in. It was not UG99, so this highlights the threat of stem rust per se. It's not just UG99 we need to be worried about. The failure we had here is we missed seeing this threat. We didn't pick it up. But the caveat here is basically we saw first indication in mid-October. By mid-November we had a full-blown epidemic. So basically your time to react with this is minimal. Stem rust is scary, just how fast it can destroy a wheat crop. But the success around that failure was that within the same season we'd worked out what this new race was. We'd got amazing collaboration. There was a paper, I think there's 20 plus authors on that from all over the world. Germplasm screening started immediately with this new race. It catalyzed advanced early warning systems, and basically that reaction wouldn't have been possible without the prior investments in UG99. So that the fact we could respond so quickly was a success. The dispersal, the early warning stuff, so this links with Cambridge University and the Met Office in the UK. This is really top-end dispersal modelling. Basically, it's now allowing us to get a handle on the probability of movements between different areas, to do forecast risk mapping, which we're just starting up in Ethiopia. So really nice collaboration, and there's some interesting things coming out of this work. So back to MLN. So this is the new one, that's the new threat that's coming up, or one of them. So basically, we've done some first assessments, but what's coming out of this, just the same as for wheat rust, is that we need effective surveillance and monitoring. Yeah, it's basically an equivalent situation to where we were in 2005 with stem rust. So I tried to put up, a few of the really important things that we needed to address with stem rust, and you can see here we've addressed most of those. But with maize MLN, then we haven't done that yet. I mean, we've got the critical focus. Yeah, that just the detection of this devastating new disease. That's our focus. USAID have now come in with some funding, and we've been able to apply the lessons we learned from stem rust, and really it's basically going to fast track the whole process. What we've learned with stem rust and what we've been able to do, we can slash the development time to get operational systems for MLN dramatically. So these investments are just so important, because these new threats are coming up, and there's going to be different threats in the future. They aren't going to go away. They're going to increase. So just to finish off, some concluding remarks. We've certainly made good progress with stem rust, without any doubt. The partnerships across the globe and the community building has been critical to this success. Those investments, we're now being able to apply them to other crops, to other diseases. But at this point, the pathogen threats are continuing to evolve. We're going to see new things in the future, and really we've got to have sustained long-term effective monitoring, the connections to the breeding programs, if we're going to keep on top of these emerging threats. We can do it. I mean, we've shown we can do it. It's challenging. It's not easy, but it's certainly possible, and I would argue that it's increasingly important. Just the threats that are coming up, the ease of movement around the globe, the concentration of the crop production, it puts us in quite a vulnerable situation. So just to acknowledge, I mean, many people have contributed to all of this. I mean, I'd flag our donors, Bill and Melinda Gates Foundation, DFID in the UK, and also USAID, but a truly global collaborative effort. Thank you.